Okay, I think we should start. The next lecture in our the course is going to be given by Kevin Cowtan. We are going to talk about model building also.
A bit about the automated model building,
okay,
yes.
Phase improvement, map improvement, averaging, and so on. And Kevin is the expert in the field, and he worked, I think, all his entire life in York. Yes?
Apart from a couple of sabbaticals in San Diego.
Okay, that is just the American, we don't count that. Okay, I think he's the expert. Just please pay attention and ask questions as much as you can. Immediately after Kevin, we will have Paul Ames, this talk on model building is good. Okay, please.
Okay, so as Gareth has mentioned, I'm Kevin Cowtan. I'm from York. Most of my career has been spent either improving or interpreting electron density maps automatically. So I'll tell you a little bit about that, and we'll probably pick up some other bits of theory that you'll need on the way. And please interrupt with questions, because I haven't met most of you, so I don't know what level that you're at, and hopefully I can say something helpful for everyone. So here's a picture of our a roadmap, really, of where we go in our structure solution. We start with data collection, processing the data, turning our image into structure factors. Experimental phasing gives us an initial electron density map, which generally isn't very interpretable. We then do some density modification or phase improvement to improve our initial phases, and that hopefully gives us a map that we can interpret, and then we build an atomic model into that map. Or the alternative pathway is, if we've got a homologous structure, then we do a molecular replacement calculation, which places a known model in our map. That gives us an alternative source of phases, which gives us an electron density map. We can go from there straight into density modification, as we would with experimental phasing. We can skip straight to model building and rebuild the model from scratch in the map that we got from molecular replacement, or we can just rebuild the model that we got from molecular replacement. It depends how close the model is, how much bias you think there's likely to be. The more steps you put in, the more bias you're likely to get rid of. But if it's very similar, you can just go straight into rebuilding and validation. So the steps I'm going to focus on are density modification, how we go from our initial poor map to a better map, and the automated part of model building. So if you've got a map like this, how do you automatically build an initial model into it? And then Paul will talk about how you go from a crude initial model to a much more complete and much more correct model. OK, so the first thing we'll talk about is density modification, going from a rubbish map to a hopefully interpretable one. And there are really two classes of techniques. There are a sort of traditional or classical techniques, of which DM is historically the most used. Parrot is my current incarnation of this. It's also in CNS and Solomon and various other software. But there's also a set of statistical density modification techniques, of which Resolve is the most widely used and is now part of the Phoenix package. And I'll cover both of those and the differences between them. But before we start on that, we really need to do a bit of background. So our starting point is that we've got some structure factor amplitudes, the strength of the spots in our diffraction pattern. That's our principal observation. But we've also got some phase estimates. Now, these may come from molecular replacement, or they may come from an experimental phasing technique, most commonly single anomalous dispersion, most commonly, again, with seleno-met derivatives. Now, these gives us different kinds of phase estimates. In the case of molecular replacement, we've got our initial model. From that, we can calculate phases from our homologous model, which will be roughly right. They will contain errors because the models aren't exactly the same. And that gives us an estimate of the phase. But because we know that it's not exactly right, we know there's some errors, what we actually get is a phase probability distribution. So this distribution can be very narrow, in which case we know the phase very well, or it can be spread out round the circle, in which case we don't know the phase very well at all. So this represents phase of about 45 degrees, real and imaginary axis in an Argand diagram. Or you can just think of phase going 0 to 360 round this circle. OK, so in the experimental phasing case, things get a bit more complicated, particularly for single anomalous dispersion, which is the most common case. We get a bimodal distribution, 2 humps. So it comes from the phasing circles. They cross at 2 places for our F plus and F minus. That means there are 2 likely phases. They're not exactly equally likely, but they're usually similarly likely. And we don't know which one is right. The phase is probably over here. It might be over here. It's certainly not over there. And this probability distribution represents that. So that's what we have coming out of our molecular replacement or our experimental phasing program. To calculate an electron density map, we need an amplitude and just 1 phase. So, but what we've got is these distributions, which are far more informative, but not so useful for calculating map. How are these represented? There are 2 different representations. One is a phase and a figure of merit. The phase is the center of this distribution, and the figure of merit tells us how wide the distribution is. If the figure of merit is 1, that means we know the phase exactly. This is a delta function, a spike. If it's 0, we know nothing about the phase. This probability distribution is equally distributed around the circle, and values in between. So this is probably about 0.5 or something. But this can only represent a unimodal distribution, phase and figure of merit. So there's a more general representation called Hendrickson-Lappmann coefficients. This is a set of 4 numbers, A, B, C, and D. And these can represent any bimodal distribution, including unimodal ones. So you could represent this distribution exactly with these 4 numbers. And so these are commonly used in experimental phasing. What do these mean? A and B represent a unimodal distribution equivalent to a phase and figure of merit. And C and D represent superimposed bimodality. So you put the 2 bits together and you can represent any bimodal distribution. So the relative size and sign of A and B and C and D control the direction of the peaks. A and B correspond effectively to sine and cosine terms. Absolute size of A squared plus B squared controls the sharpness. This is closely related to figure of merit. So for molecular replacement, because we had a unimodal distribution, we get A and B with C and D equal to 0. So in that case, we've got a unimodal distribution. If you see C equals D equals 0, then you know it's unimodal. When you combine them together, we can describe any bimodal distribution. So if the A and B peak were shifted this way a bit and combine that with a C and D like that, then you get something like this. It's actually the product of the 2 distributions. So it's a little bit more complicated, but that's the general idea. Right, so that's our starting point. That's what we have when we get to the point of calculating our first map. And how do we calculate a map? Well, we need just 1 phase. So what we do is calculate the electron density map, which is least noisy with respect to the uncertainty in the phase. And it turns out there's a very simple way of doing this. If we take the center of mass of this phase distribution, here it's going to be about here. It's not going to be out here because the phase is spread a bit. It's going to be in, well, probably just in just a little bit. As the phase gets more spread out, as our uncertainty increases, it moves down to the center. So everything gets down-weighted the more uncertain we are. That's the natural way of doing it, and it gives what is the least noisy map. In this case, you can see the center of mass of the phase distribution would be about here, so it gets weighted down a lot more. If we could eliminate this second peak, we'd get a lot more confidence. So that's how we calculate our first electron density map. Problem is, that's going to be noisy, especially in the SAD case, because we've got this split between these two peaks, meaning that this is weighted down a lot. So we would like to improve our electron density map. And this is where density modification comes in. It's really a problem of combining information. So we start from our reciprocal space phase probability distribution. And we want to put in some information in real space to try and narrow down the possible phases. And it's really a problem of combining information in reciprocal space, where we've got our structure factors and our phases, and in real space, where we have an electron density map. So how does this work? Well, here's the most rudimentary picture. We start with a structure factor amplitude and a phase. We calculate an electron density map using a Fourier transform. What we're going to do is modify our electron density map. We're going to try and, it's going to be noisy. We'll try and reduce the noise a bit using basic image processing algorithms. And then we will calculate an inverse Fourier transform. And that gives us a new set of structure factors, amplitudes and phases. Both the amplitude and the phase will have changed. Now, then we can use this information. In particular, we could just substitute the phase in for our original phase and keep the observed amplitudes and start the process all over again. And if you're close to the answer, even that very, very simple approach actually works. It's a bit rudimentary. We can do a lot better. So, but that's conceptually what goes on in a classical density modification calculation. In practice, we're a bit cleverer. We use some sort of combination of the experimental and the modified phase. Now, I mentioned that we're really dealing with phase probability distributions, not individual phases. So what we actually do is start with our phase probability distribution. We calculate our center of mass of that distribution, as I described earlier. That gives us a weighted F and a best phase. And that's what we calculate the map from. And when we do our back transform, that gives us an amplitude and a single phase, not a distribution. We then turn this into a distribution again, and then we can combine the original phase probability distribution and the new one by multiplication. How does this work? Well, basically, we look at how well this calculation reproduces the observed Fs. And that's an indication of how big the errors are in these phases. And that allows us to turn an individual phase into a probability distribution about that phase. Once we've got an error estimate, we can do that. OK, there's a flaw in, fairly fundamental flaw in this approach, although it's better than what I've shown you so far. You can only multiply probabilities if the two probabilities are independent. So 2 die rolls, they're independent. So 2 6s is 1 in 36. The probability of a father and son both having fair hair, they're not independent, so it wouldn't be the product. So there's a problem. And in fact, this probability distribution is very strongly dependent on this, because we got it through this cycle from the original one. So there is a tendency towards bias in this calculation. You run this calculation over and over again, and all your figures of merit creep up towards 1 without the errors going towards 0. So the figures of merit are overestimated. You've got bias. How do you deal with that? Well, there's a very clever method by Jan-Peter Abrahams, which mitigates it partially. It helps a lot, although it's not a complete solution. which is to add an extra step in here called gamma correction, where you subtract out some of your initial signal and you subtract out the right amount to remove the bias. And this is the method that's implemented in DM and Solomon and CNS. There's been one more recent development which goes into my program Parrot, which combines these steps that were down here into a single step using the same kind of mathematics that's in Garib's program RefMac, and that improves things a bit further. It just throws a bit more information at the problem. So that's probably there's one further development in the pipeline coming from the Leiden group, but that's the state of the art for most common purposes. Now, the other approach that I mentioned is the statistical method, which we'll look at a bit later on, which is a very different approach in that we break the connection here. We do calculate a map, and then what we do is infer a probability distribution for the densities at any point in the map. And then we carry the probability distribution into reciprocal space and then do the multiplication here. Because there's a much weaker connection here, this has less problems with bias, although there's not much difference between the two approaches these days. I'll show you how that works a bit later. Right, so that's conceptually how the calculation works. We can look at a few of the details. And the most important thing is what we do to our electron density map to improve it. It's basically an image processing problem. It's the sort of thing you can do with your photographs, but done in 3D and with a bit more crystallographic intelligence in that. So there are three very widely used techniques which have proved successful and widely applicable over time. There's been a few more experimented with, but none have been as effective as these. So the first is solvent flattening, second is histogram matching, and the third is non-crystallographic symmetry averaging. And the aim of all of these is to go from an initial electron density map to one which is more realistic. So solvent flattening is the oldest and probably the most powerful on its own. Here's a two-dimensional section from an electron density map, contoured, well, a stack of a few sections. And you can see just from this map that we've got regions where there are lots of features and regions where the features are rather sparse. And you would probably correctly deduce that this corresponds to protein and this corresponds to solvent. And the solvent is disordered, and therefore we don't see much in there. So from this, you could draw a mask, an envelope, dividing the unit cell into protein regions and solvent regions. And that's fairly easy, even from a fairly poor initial map, and is done completely automatically. So the question is, what are the features in the solvent? Well, if your phases are very good, then they correspond to ordered solvent, real features in where the solvent isn't completely disordered. If your phases are very bad, then you also have a load of noise in there, and in practice the noise tends to swamp any real signal. So most of the features in your solvent early in the calculation are actually noise. They're not ordered waters. So because of that, if you just flatten out the solvent, then you're getting rid of the ordered waters, the ordered peaks, but you're also getting rid of the noise. And when your phases are poor, you're getting rid of a lot more noise than signal. And as a result, on aggregate, the map has improved. So you flatten the solvent, you end up with an improved map from which you get a less noisy map, from which you get less noisy phases, which then feed back into the next cycle of the calculation. So that's a very simple technique. It works. It's been proven over a couple of decades now. Now, it would be nice to be able to do something similar to the protein region, because that reduces the noise in our solvent. What can we do with the protein? So there's a technique from image processing called histogram matching. And this is based on the observation that noise maps with random phases have a Gaussian histogram. You have a mean value of the electron density, which is the most common value in the map, and you have decreasing proportions of more extreme values at the high and low end. However, a well-phased electron density map, one which isn't noisy, has a very different distribution. It's very skewed. So you have a few grid cells which have high densities corresponding to atomic peaks, and a lot which are fairly low, and very few which are negative due to resolution truncation. So, and this corresponds to the gaps between the atoms, and these are the atomic peaks. So what we can do is sharpen the electron density in the protein region using a transformation which matches our initial histogram to what we expect it should be. And this is histogram matching. Here's a little picture if you're interested of how it works. We just apply a monotonically increasing curve which transforms one histogram into the other whilst keeping all the points in order. That's how it works. And you can find similar features in your photo processing software. And this works well. It's effective at better than 4 angstroms resolution. So those are very simple, something we can do to the solvent, something we can do to the protein, and they're complementary, and we end up with the whole map being improved on aggregate, and so we end up with better phases. Now the third and slightly more complex technique is non-crystallographic symmetry averaging. So if a molecule has internal symmetry within the asymmetric unit, we can average together the related regions. So here's an insulin dimer. It forms a hexamer, but this particular insulin with 3-fold crystallographic symmetry, and then two molecules within each asymmetric unit related by a non-crystallographic symmetry average. We'll talk about the distinction in a moment. If we just average the electron density of these two together, the bits which agree, the real signal, will reinforce each other. The noise will sometimes reinforce and sometimes cancel. So we improve our signal-to-noise ratio by averaging them together. It improves by √2 in theory. In practice, if a full density modification calculation is performed, recycling through reciprocal space, as I described earlier, that forms powerful phase relationships. So actually the improvement that you get is rather better than that √2 might suggest. And in theory, with 4-fold non-crystallographic symmetry, we can phase from random. In practice, you can't do that because you need to know stuff that you don't know. But in theory, the information is there if you had a way of getting at it. We've done toy calculations that prove that. OK, so there's some important terminology here that we need to understand. Firstly, the distinction between crystallographic and non-crystallographic symmetry. Now, crystallographic symmetry maps the whole crystal onto itself. So here's an example, 2-fold axis aligned along a unit cell direction. It matches this copy onto this copy. So this is something like P2. It maps this edge of the cell onto this edge, this corner onto this corner. It matches these two onto another 2 here, and these 2 onto another 2 here, and it maps this one onto this one. So it's global. The whole of the crystal obeys this one symmetry operator. Here's another example, a 6-fold aligned perpendicular to this plane. It maps this copy onto this copy, this onto here, but also maps this one onto this one, and this one onto this one, and this one onto one down here, and so on. So again, it's global. Non-crystallographic symmetry, the important distinction is that it's local. So here we are. Here's a twofold which isn't aligned with the unit cell axes. So it maps this molecule onto this molecule. The same axis maps this molecule onto itself about here. So it would clash with itself if we flip this over using this axis. And if we apply the symmetry axis to the lattice, this lattice here, this rectangle becomes this one, which doesn't match at all. So it's only obeyed over a limited region within a single asymmetric unit. So it's a local symmetry within the asymmetric unit and doesn't match over the whole crystal. So here's another example, a five-fold axis that must be non-crystallographic because there's no way a lattice can contain a five-fold symmetry. So non-crystallographic symmetry is local, crystallographic symmetry is global. Some more terminology. You may come across proper and improper NCS, also described as closed for proper and open for improper. A bit of symmetry terminology. So in proper or closed NCS, every operator maps a copy of the molecule onto another copy. So a twofold axis here maps this copy onto this copy, but it also maps this copy back onto this copy. And that's because in this case, it's a perfect twofold rotation. Here's a different case. In this case, it's a rotation of I don't know, 70 degrees or something. This maps this copy onto here, and this copy into Solvent. So it doesn't form a closed group. It maps A onto B, but B onto nothing, rather than A onto B and B onto A. So anything that's not a pure rotation is most likely an improper NCS or open. There are a few exceptions, but that's your general rule of thumb. So that's one piece of terminology. Multi-domain averaging. This is a problem that occurs when you've got a flexible protein. So here we've got A and B linked by a hinge. You can see A is related to a copy of A by a twofold rotation. B, the hinge is in different conformations. So the rotation between B and B is not 180 degrees. It's a bit different. So then you would need a different relationship between A and between B and B. And you call A and B domains. And you have to use multi-domain averaging where these are averaged separately. The final case is multi-crystal averaging, where you've got multiple crystal forms, and you can employ NCS averaging in that case as well, and it's very powerful when you can do it, although it's a much harder calculation. So here's an illustration. We've got two crystal forms. This has two NCS copies. This one has one NCS copy. This one has phases. This one doesn't. We can, if we can work out how to do the calculation, which is challenging but can be done, then you can average between all three copies and you get a lot of phase information out of it. But that's advanced, not for the faint-hearted. How do you know if you have non-crystallographic symmetry? Well, cell content analysis is the principal method. How many monomers are there in the asymmetric unit? And if you run Parrot, it will give you a little table of what it thinks the cell content is. You give it the sequence, it works out how much space that sequence would take in the unit cell, and if that would give a ridiculously high solvent content, then your best guess is that there are multiple copies, and Parrot will give you a table of how likely different numbers of copies are. And you may have some external information which may feed into that as well. You may know that it forms a trimer, for example. You can do a self-rotation function. That's a molecular replacement of the data against itself. And you may be able to detect rotations like that. Also, difference patterns can detect pseudo-translations where the molecule moves without rotating. How do you determine the non-crystallographic symmetry? Well, there are two things we need to know. We need to know the operator, the rotation and translation, so that we can average the density together. And we need to know a mask. Because the NCS is local, we need to know where in the unit cell it applies. We can't average everything together because we'll average stuff which doesn't match up. So the operators are most commonly determined from heavy atoms. And if you have 3 heavy atoms in each copy, then by matching similar triangles, then you can find the NCS. It's not a foolproof method, and it varies depending on the number of heavy atoms that you have, seleniums or whatever, but it's the most automatic. From initial model building, if you've got a good enough map to build at least some fragments, say some helices, then Parrot will match those up automatically and deduce your NCS. From molecular replacement, as long as you found every copy, then you get it automatically. There's a couple of other methods. There's a method in Phoenix called NCS from map, which is slow, but it's a good fallback if nothing else has worked. Or the other thing you can do is, if you think you know where a molecule is by looking where the density is, where there's a big lump of high density or strongly varying density, then you can draw a mask around that and feed that into a molecular replacement calculation. Again, that's a desperate measure for advanced users. The mask, once you've got the operators, the mask tends to be determined automatically. Parrot will do that for you. And it does that by looking for regions which obey where the agreement of the rotated densities is good. Right, so I think I've got a picture of user interface. In practice, in all the standard cases, doing it is very easy. What you do is just give it your MTZ file, your structure factors, and your sequence, and it will do normal density modification. If you want it to do NCS averaging, you give it your heavy atoms or a molecular replacement model, and it tries to work out the NCS for you and do everything automatically. So whilst the concepts can be complicated and sorting out the difficult cases can be complicated, in the easy case, it's automatic. You just tell it what you know and it does its best. Right. Do we want to see? This is a slightly more advanced concept, but it has relation to refinement, so I'll mention it briefly, and the more you hear it, the more you'll understand it, hopefully. How do we go from a amplitude and phase that we get from our modified map, once we've finished improving it, to a phase probability distribution? Well, this is a likelihood calculation, and you'll hear about lots of those in crystallography. And the basic concept is that we've got our known structure factor amplitude, and we've got a structure factor amplitude and phase which contain errors, which come from our modified map. Conceptually, they could come from an incomplete model, but in practice, in our case, they're coming from a map with noise. Now, if the error term here has a phase which is independent of the phase of our current map, then we'd expect the phase errors and the magnitude errors to be equal in size, because this could be distributed randomly around a circle when we average over a lot of structure factors. So if we average over a lot of structure factors, the discrepancy in the magnitudes here is an indication of the average discrepancy in phase over a lot of structure factors. So that's how we estimate the uncertainty in our structure factors. There is a slight point of well, it confused me for a while. You would have thought that you can always fit the observations better by simply introducing a bigger error term. So That's what this little animation is about. It shows that because the probabilities add up to 1, as you increase your error term, your Gaussian gets wider and the top gets lower. So, yes. So the area is constant.
Good description
Yep.
of probabilities.
So as we spread out this Gaussian, its overlap with the observations increases. Here we are. You can see it spreading here, and it goes up, and then it goes down again because the center's going down. So it goes up as it gets wider, then goes down again. And that means we can get our maximum likelihood estimate of the uncertainties. So that's one of the that's how likelihood allows us to measure, to estimate not just parameters but uncertainties in those parameters. And your uncertainties are always a vital part of your knowledge, so that's important. I don't think there's anything else. We'll skip the rest of that. Okay, well, I'll mention one more complication that arises. So when you're doing NGS averaging, so this is a new feature that's in Parrot primarily, which is that when you are combining NGS-related copies, sometimes it gets messy because the molecules are the same in regions and different in regions. So here I've got a trimer, and each trimer has 3 domains, A, B, and C. And the B domains are conserved. The A domain is conserved between these 2 copies, and the C domain is conserved between these 2, but this A and this C have been distorted a bit due to crystal contacts or something. So how are we going to do our averaging? We can average all the B's together, that's fine, and we can average BC between these 2, and we can average A between these 2, but we can't average these 2 with everything. So this used to be a big pain, but in Parrot it's just handled automatically. It calculates a separate mask for pairwise for every single pairwise relationship. And so it just handles it for you. It does mean with high order NCS, Parrot gets a bit slow, but you can fool it by only giving it some of your NCS, and then it works anyway. So that's another problem case, automated. Here's what it looks like. So normally you just give it your sequence and your MTZ. It picks up the columns. You have to tell it your Hendricks and Lattman coefficients. You can click this if you've got phase and figure of merit instead. And then you just click run and it works. If you've got non-crystallographic symmetry, then you can click this to tell it your heavy atoms, or click this to tell it a molecular replacement model or a partial model if you've done some initial model building, and it gets the NCS from those. In that case, you may want to increase the number of cycles of phase improvement. It runs 3 by default, which is a good compromise between improving the maps as much as possible without overly biasing them. However, if you've got NCS, then you can increase that. I usually start with 10. There are extreme cases where you can run for hundreds of cycles and get an improvement, but it's such a quick calculation that you can just try it and look at the maps. Right. Now, I mentioned that there was a second class of techniques, statistical density modification. The only one which is in widespread use is Resolve, which is also in Phoenix Auto Build. And I'll give you a brief overview of how that works. That's not a very good picture. So the way this works is we form a statistical distribution of expected map features. Now, I've already said we can determine from even a very poor map where the protein and solvent are. Now, we expect the protein region, on the basis of looking at hundreds of maps, to have a higher variation, because it's ordered, and a slightly higher mean. Whereas the solvent, mostly oxygens and 2 hydrogens for each one, has a lower mean, lower density on average, and because it's disordered, it shows less variation. So that's a statistical description of what we would expect the electron density to be like, given that we just know the protein and solvent boundaries. So that means that if someone gives us an electron density map, say this yellow one, we can say, actually, this looks quite a plausible map because it has more variation in the protein, less in the solvent, higher mean in the protein, lower mean in the solvent. So it looks plausible, and we can turn this into a probability and give it a number, how probable this map is. If someone gives us this orange map, we can do the same calculation, and it will immediately tell us that this map is improbable. Now, every electron density map corresponds to a set of structure factor amplitudes and phases. So this yellow map corresponds to this structure factor, this orange one to this one, and because this map was probable, we can say this structure factor is probable. And similarly, we can say this one is improbable. Now that means we can turn our electron density map into a probability distribution over all the structure factors. All of them interconnect, which is a slight complication, but we can do it. That we can just multiply by our original phase probability distribution and get an updated distribution. So conceptually, it's actually simpler. In terms of implementation, it's a bit of a nightmare, but you can do it. It's very clever. So that's used in Resolve. The methods go back to Gerard Bracoin, and it's slow, but because it's such a different method, it probably on average performance is about the same as the latest classical methods, but it sometimes works in different cases. So it's complementary and worth a try. There we are. Okay. That's of historical interest only. I'll just show you one quick example. This is 50-odd structures where I ran density modification with and without, in the latest methods, with and without non-crystallographic symmetry. And you can see some structures don't have any non-crystallographic symmetry, so it makes no difference. Where you do have it, then, so this is agreement without NCS and with NCS, with one representing perfect. And so anything above the line, NCS has helped us. So here we've got a dramatic improvement in the quality of the map. Here we've got a more minor improvement. The ones on the line, there was no NCS. Here it detected it wrongly, but 1 in 50 chance of that. So it's not a huge problem. Right. OK, so that's our overview of density modification. Any questions about that? Yep.
I have one question about NCS.
Yep.
Usually when you have a molecular replacement or something, you have one part of structure you already know, and what you are interested in another part.
Yes.
And masks may only have information about the part you know.
Yes.
And when you do averaging, and then parts you are interested in may be suppressed. Is there a way of dealing with that?
Actually, the mask tends to grow over multiple cycles to include, particularly in Parrot, as the mask isn't binary, it's not 1, 0. It does tend, the phase information and along with the observed amplitudes tend to pull you towards the true map, which means that features start to appear in the related but unmodeled regions, which means the mask tends to grow to include them. So in practice, while it's a problem on a single cycle, but when you run an iterated calculation, it tends to do the right thing. Of course, regions which are disordered between the two don't tend to get rescued that way, but then maybe you're interested in the disorder.
That you may come to that later.
Yep. Yep, but actually, for dealing with that sort of problem, the really difficult bits, okay, this is a fundamental point. Density modification is getting you to a point where you can start your model building. And once you've got an initial coordinate model, that's a stronger constraint on the phases than density modification. So the problem areas you generally go back to in your refinement and rebuilding cycle, and you'll get more information at that stage than you will at the density modification stage. So the fundamental point of density modification is getting you your initial model or your initial rebuild, not filling in the final details, because the refinement is a stronger density modification. Okay? Right, I'll talk briefly about model building, the automated model building that you do offline, and then that leads into what Paul will talk about as to building the bits that don't get built automatically and fixing the bits that are wrong. So for proteins, we've got a variety of software: Buccaneer, ArtWarp, Phoenix Auto Build, which incorporates Resolve. And then for nucleic acids, there's Nautilus, most of which is also incorporated in Coot, ArtWarp, and Phoenix Auto Build. So we can auto build proteins, nucleotides, sugars are on their way. My protein building software is Buccaneer, which it's specifically developed to be good across a broad range of resolutions. How does it work? Well, the problem I was trying to tackle with this was that electron density maps look very different at different resolutions. So if you build any knowledge about what a electron density map should look like into your program, then it only works at the resolutions where that knowledge is valid. So I tried to get around this problem by being a bit clever about what I'm looking for. So what we do is we start with the map that you're trying to interpret, your work structure. And what we do is we generate a reference structure, a map for a reference structure, which has the same resolution and the same level of noise. We take a known structure. It's just a standard library structure. You don't need to select a homologous one. And we make a set of structure factors and phases which are just as rubbish as the ones you're trying to build from. So then we know what the electron density corresponding to any atomic feature in this map looks like. So when we see similar features in this map, we infer that the corresponding atomic features are similar. So in practice, this works where we use a C alpha target for detecting regions around a C alpha. It's in a 4 angstrom sphere around a C alpha. We look for specific high and low density features. Well, it's all done automatically with a likelihood calculation, but in practice, what it ends up looking for is conserved high and low density features. So for the region around a C alpha, we expect to see high density in the blue areas, but we also expect to see a low density in the red areas. And these are actually the more powerful constraint on whether we found a C alpha or not. So that can be turned into a search target, and you can search your electron density map and find C alpha positions. So that's one of the targets we use. We also use a series of 20 targets based on the C beta position for identifying sidechain types. Again, with the same likelihood calculation. So this goes into a cyclic calculation, each cycle having 10 stages in which we find C alpha positions, grow them into chains, join and merge the fragments, link termini, doctor sequence, correct for insertions and deletions. There's a filtering on pore density, rebuild NCS, that's very useful, prune any remaining clashes, and then add on the side chains. So I'll show you a picture of what this looks like for, this is a 2.9 angstrom map, and this is an awkward loop. And the first step is to find C alphas. So just using that likelihood target, it thinks this is a C alpha position because the density is the right shape. It thinks there's another one down here, another one down here. Can we see one up there? No, I don't think we can. So that's its starting point. Next, it uses Ramachandran restraints to try and build additional C alphas on either end of these. So it tries to build peptide bonds to the next C alpha and score those. So that gives us lots and lots of chain fragments. So starting from each seed position, it's built lots of fragments, and some of them are redundant, containing the same information, and some of them are contradictory. But you can see we've got a helix here. We've got two choices of how to bridge this gap. We've got a bit going off over there. Right, so the next step is to merge the ones which are telling us the same thing. So that gives us that. Everything merges here. Where we had a choice of routes here, we always take the longer one because that means we get the helices right. Otherwise, we take shortcuts down the middle. We couldn't resolve this one because it's running in the opposite direction and it branches off. But that's the best we can do at this stage. We can't resolve that yet. So next, we apply our 20 sidechain targets and work out what the likely sidechains are on each C alpha, and then slide that against the sequence and work out if there's a way that can be matched up to the sequence. And that's what we got in this case. Here, it found that it couldn't get a good match to the sequence straight off. But if it inserted a dummy symbol in the sequence here, then it could get a good match. So that was the best match. That suggests we've built one too many residues somewhere in this region, not necessarily here, but within the vicinity. In fact, it was here, as we'll see. So then whenever there's an insertion or a deletion, you try to rebuild to eliminate it. So it takes sets of 3 residues and tries to replace them with 2. And what that did would found the shortcut across here, and it thinks that's more consistent with the sequence. Any remaining clashing chains are pruned, with long chains taking precedence over short and sequence taking precedence over unsequenced. So this one's selected as the favorable one, and then we build side chains, and here's what the final refined model looked like, and we've got the basic structure right. So these 2 side chains, because the main chain is a bit out, are wrongly positioned. But this then gets iterated with refinement, and many of the problems are sorted out. So that's the basic process. Here's a little illustration of 50 structures again, which I tried this on. Resolutions 1.4 to 3.2. Some of them it built the whole lot. Even at 3.2, it managed at 80% of this one. We've seen successful structures down to 3.7 angstroms, and the Leiden group have got it working down as low as 4 angstroms, but that involves some additional methods as well. There are some methods, some cases where it doesn't work at all well. What's going on there? Well, if we look at the dependence on the initial phases, so this is a measure of how good the phases are, good phases, bad phases. You can see that it all depends on the initial map correlation. If your phases are good enough, then it works. If they're not, then it doesn't.
Is there a jump there, or it is just the absence of the?
It is a jump, yes. Yeah. And this is something that the Phoenix and ArtWarp people have found as well. Map correlation between 0.5 and 0.6 is the point where it fails, although ArtWarp can bootstrap from poor phases if the resolution is very good. How do you use it? Very easy. Again, it's just like running Parrot. You just give it your sequence and your MTZ and click run, and it runs and builds it. It's pretty quick. It's quicker than ArtWarp and much, much quicker than Phoenix. Having said that, the methods are significantly complementary. ArtWarp in particular is stronger at higher resolution, but weaker at lower resolution. So if you've got good resolution, lousy phases, then ArtWarp is more likely to work. Poor resolution, better phases, Buccaneer is likely to work. they're both automatic, so try them both. What comes out is a fairly, well, it may not be a very complete model, and it will have things which need tidying up. You can tidy it up in Coot, or actually you can use it as a preprocessor to ARPwarp, and actually doing that, you get a better result faster than just running either program on its own. Typically, things that you might have to do in Coot, it tries to connect up the broken chains now, but you may have to do some of that by hand. Extensive use of density fit and rotor analysis to look at dodgy residues, check the Ramachandran mole probity, add waters, ligands, look for unmodeled blobs, check that the chains are complete. Are there any residues missing on the end or unsequenced at the end? They'll be marked as residue type UNC for unknown. Occasionally, you also get extra residues built on the end which aren't in the sequence. Then you need to check your sequence, but usually it means it's just been too enthusiastic. Re-refine and examine the difference maps. So here was a difficult case. You've got this resolution is only 3.6, and you've got this long helix which Buccaneer built completely correctly automatically, and then the bit at the end is actually the really challenging bit, and Buccaneer built enough to get a starting point on this. Okay, and the other really new tool is Nautilus, which is the nucleotide building program. There's an automated version in CCP4i. It runs just like Buccaneer. Or interactively, you can run it in Coot. It won't dock the sequence in Coot, but it will build chunks of nucleotide near the cursor. So I think it's obvious what it's trying to do. It's a related method to the method in Buccaneer. It's looking for consistent high and low density features which indicate the presence of either a sugar or a phosphorus. Here's an example of the Coot version. So here we've got some bases stacked, and what we'll do is we'll just click the build nucleic acid button and ping. There it was. Okay, so here it's done clearly a pretty nice job. It hasn't assigned the base types. For that, you need the offline version. Here, actually, it's got it wrong in the other half. That's nonsense. It's tried to build it sort of in the wrong direction. So what should we do? Let's go over here and click the button again, and it'll build some more. There we are. And it's actually overwritten the wrong stuff that it built earlier. It's completely replaced that. And we can see that now looks a lot more sensible. And the new bit's been joined onto the old bit because there's a hairpin up here. And there you can see it looks fairly sensible. And we can have a look at the density here. And you can see that actually it's pretty broken. If this were protein, you couldn't build it so easily. Here it's a bit clearer, but there's still gaps in it. But it's clearly right at this point. Here it gets a little bit more confusing. I'm not experienced enough to be able to build this bit right, but if we turn on the rest of the atoms, then you can see it makes perfect sense as an explanation of the density. You can I'm working on a paper describing all of this, but the presentation's also on the website, so you can have a look at that if you're interested in the details. That just is an indication of the sugar and phosphate targets, with blue and red crosses indicating places where you'd expect high or low density. But that's just an illustration of how we assign the sequence. There's a set of probe points which are indications of whether we've got A, C, G, and U. We can't tell C from U. We could if it were DNA. I haven't exploited that information yet. But we can certainly distinguish large and small bases, and we can sometimes distinguish between the types. And so that gives us partial information. But when you try reconciling our partial information, we can't tell C's from U's, so these are always the same size, and we can't strongly tell G's from A's. But when we try and reconcile what information we have with the sequence information, here we're strongly confident it's an A or a G, so no, yes, yes. But if we want to match all of them up, we can see that only this one makes a lot of sense. There we are, because everything's high probability. So that works well. I've just been doing some comparisons with ARP Warp and Phoenix Refine. It certainly works. We've got a user report on a ribosomal protein at 3.8 angstroms. My experience is that it's much faster and much better than the equivalent tool in ARP Warp. It's comparable in terms of completeness of the final model to the tool in Phoenix, but it's about 100 times faster. So, but the Phoenix does a brilliant job of refining it. So for my ultimate nucleotide strategy is run Buccaneer as a preprocessor to Phoenix. So my ultimate protein strategy is Buccaneer as a preprocessor to ARP Warp. So different combined strategies for different problems. I've seen one structure which it can't cope with at all, which is a quadruplex structure. So maybe there's more work to be done there. A few acknowledgements. The JCSG data archive is invaluable. Actually, also the work of the Richardsons on cataloging good protein and RNA structures, that's coming useful. Garib, Raj, and Pavel have offered a lot of help over the years, as have Eleanor and Paul, Randy Reed, Clemens. Funding has come from the Royal Society and the BBSRC.
Thank you. It was very, very good and excellent lecture, and especially last bit, a lot of people are very worried that there are no tools for nuclear RNA, DNA, especially in LMBIM. If it would be earlier, few years, then some people would have done ribosome structure probably a bit faster. But nevertheless, please, questions?
Yep.
For this Nautilus, do you input any kind of restraints on the kind of base pairs? Or not, all this information is completely out?
No, you only give it the sequence. So that gives you a validation, actually. If it sequences two the two sides of a helix in a way which is inconsistent, then that's telling you that it's got something wrong. But it doesn't use that as a constraint. No.
I think after that you could do refinement. In refinement, you could use base pair restraints, and then you could come back. We should talk about that later. How to do it.
So will Buccaneer try to build, if there's locally no density for one or two residues?
So it will build across, if it has an N and a C terminus with sequence numbers, because both ends have been sequenced, which support a short link, then it will automatically build that link if there is a plausible way of doing so. If there is only one sequence number missing. There is a feature which hasn't been released which will cope with longer loops, but I need more intelligence in the control software to work out when to turn that feature off. That exists in CCP4 as a separate program called Sloop, which you can run if you're confident it's time to do that. But it's not turned on in Buccaneer by default.
Any more questions? If not, then thank you again, Kevin. As I said, that we will have a second lecture.